Good morning and welcome to another lecture for Bio 110. We're going to continue to explore in this lecture how membrane transport works within the cell and look at some of the components that are going to work in the depolarization of membranes in neurons. So in the last lecture, we learned about active transport. We learned about ion pumps, for example, the sodium, potassium, the calcium, and proton channels. We also learned that those pumps require energy, and that energy can come from ATP hydrolysis or light to be able to move those ions across their concentration gradients. We also learned about ion channels, that they're selective and that they could be gated or non-gated, meaning leaky, so they could open up or close. We also explore some of the qualities of some of the ions, like potassium. And what we're going to do today is to now look more closely at some of these ion channels and the effects that they have on the biology of the cell. But before we start with this topic, I would like to show you and discuss with you how we study some of these channels, and that's going to be using the patch clamp technique. The patch clamp technique has been used in neurobiology for many years, and it involves the taking of a glass tube, a microelectrode, which has been generated to be about a micron in size, so it's very small. And with that microelectron, what we can do is to create a tight seal around the membrane of the cell that we want to study. In the membrane, as you know, we're going to have the channels or the pumps that we want to study. Now, since we have a tight seal across the membrane, we can basically pierce out that small piece of the membrane, taking with us the receptors which are present. Here in the right, what we have is an image of a neuron cell which has been treated with the patch clamp technique. We have a suction pipette that is keeping the neuron in place, and with the recording microelectrode, the glass tube that I mentioned to you, we are able to take a small membrane piece of that neuron cell to study it. Now that we have that small piece of membrane, we'll put a buffer inside of it. So we have a condition with a buffer that contains a defined concentration of ions, and we can then put it in a tube, for example, that is also going to have a buffer with different ionic conditions. What we also have is that we can connect the entire system to an oscilloscope, and that oscilloscope will have an electrode that is present in the media that is going to be in the cytoplasmic side of the membrane, and we're going to have another electrode that is going to be present inside the fluid of the microelectrode. And therefore, whenever there is an opening of the channel, we can measure the current that flows as the ions are flowing from one side of the membrane to the next. And that is registered through the oscilloscopes as peaks that indicate that the channel has opened and fired. So for example, what we have in here, it's a graph against time where we're measuring the current in picoamperes, so very small current opening. And what we have is that whenever the channel is closed, the current is zero. So you have a oscillation in the current that is marking at zero. With time, for example, at time 7 milliseconds, we can give a stimulus to the channel, and that now is translated as the opening of the channel and an increase in the current within the system. And that then, when the channel closes, the current go back to zero. So every time that the channel allows the flux of ions across the membrane, there is going to be a current that is going to be measured by the oscilloscope. And therefore, at this point, what we can do is to modify the conditions to see under which conditions the channel is able to open, and the direction also of the current. If the ions are moving from one side of the, for example, the cytosolic side of the membrane to the external part of the membrane, or if the ions are flowing from the external part of the membrane into what used to be the cytosolic side of the membrane, and that will give you a different level of current. Now that we have a way to measure the current across a membrane by the opening or closing of the channels, we can then study and define membrane potential. So what we're going to look is to the definition of the membrane potential, how the membrane potential is generated, how it is measured, and then look at the particular terms that are important to understand these principles: the resting potential, the action potential, and depolarization. And we're going to see that the effect of voltage-gated ion channels are going to be the ones that, once open, they are going to induce changes in membrane potential. Now, just as a review, let us look at the electrochemical gradient. So we talked before that if you have a membrane, and this membrane here, it's displaying a concentration of an ion that is greater in the outside than the inside, you do not have a membrane potential because the ions cannot move across the membrane. But if the ions are able then to move across the membrane, they will move and now affect the charge of the membrane around it. So you will have an electrochemical gradient that will be a lot greater because the membrane potential is present, and therefore these ions, which will have a tendency to move across the membrane due to their gradient, now also have the force of the membrane potential that is going to help them move towards the positive sides. That is going to make them move towards the negative side of the membrane and be repulsed, by the positive sides in the outside. So the electrochemical gradient, as you see by the arrow being thicker and bigger, it's stronger. Now, in this other case, we have the example of an gradient, which is, again, it is greater in the outside than the inside, but the direction of the membrane potential has switched, and therefore the negative ions are outside the membrane and the positive ions are inside the membrane. Since the membrane potential is working against the direction of the gradient, the electrochemical gradient for this particular ion, it's smaller. But still, it's able to allow the movement of those ions across the membrane to the inside of the cell. So again, the electrochemical gradient is the combination of the membrane potential and the chemical gradient, and they are formed at different concentrations of ions. So remember that the membrane potential is a component of the electrochemical gradient, and that is the electrical voltage that you have across the membrane based on the concentration of ions that are present across the membrane. Now, let's look at the membrane potential and how the membrane potential is established. So in this image, what we have, it's a membrane that it's dividing two different environments. Every plus sign in either side of the membrane, it's indicative of a positive ion, and every negative sign in either side of the membrane is indicative of an anion. When you have an exact balance of charges on each side of the membrane, since both positive and negative are balanced and equal, the membrane potential of that system is 0. However, when now you have an imbalance in the membrane potential because you have more positive ions in one side of the membrane or more negative ions in the other side of the membrane, that has generated now a membrane potential that is non-zero. And that is the example on the right. So a few positive ions that have crossed the membrane from the right of the membrane environment to the left have set out now a non-zero membrane potential, and therefore, as you can appreciate, the charge to the left of the membrane is slightly positive and the charge to the right of the membrane is slightly negative. So how is this established? Or how it is measured? Let's just start first with the measurement point. So we can measure the resting membrane potential by using a potentiometer. And a potentiometer is a small instrument that allows us to put a microelectrode and that microelectrode can be inserted into the cytoplasmic side of the cell, and of course it's going to have a conducting buffer inside of it, and then we can have a reference electrode that is contacting the medium of the cell outside of the plasma membrane, and therefore we can observe the change in voltage across the membrane by measuring the ions that are present in each side. Now, let's take an observation of a potentiometer in an artificial environment. Here what we have, it's a flask that has a membrane, shown here by this blue arrow, that is separating two environments. One of them environment artificially resembling the cytosol or the cytoplasm, and the other environment representing the extracellular medium of the cell. As you can appreciate, the concentration of sodium chloride inside the cell was set up to be 15 millimolar, and the extracellular concentration of sodium chloride in this on the other side, in the extracellular medium, was set up to be 150 millimolar. So you have a greater concentration of sodium ions in the right side of the membrane than in the left side of the membrane. Conversely, the concentration of potassium chloride inside the cell was set up to be 150 millimolar, and the concentration of potassium chloride in the extracellular medium was set up to be 15 millimolar. So a much greater concentration of potassium ions in the area that's designed to represent the inside of the cell versus the outside of the cell. Since the ions do not have a way to cross the membrane, they can be considered to be at equilibrium, and therefore the membrane potential is zero. So the ions do not move. But if we now enable one type of ion to move, in this case the sodium ions, we can put a channel in the middle of the membrane. Again, we have the same concentrations in what is represented to be the cytosol versus the extracellular portion. And therefore we allow the ions to move and separate across the membrane. So what you can appreciate is that the positive sodium ions are able to move from one side of the membrane, from the right, to the left. That is now going to set up a current, and that current will be measured by the potentiometer as a electrical membrane potential of minus 59 millivolts, because the ions are moving from the extracellular quote-unquote region to the intracellular quote-unquote region. So the ion movement eventually will stop because you will reach the equilibrium of sodium ions, meaning the conditions in which the gradient of sodium ions equate the pull of those ions by the membrane potential established across the membrane. So you have also the repulsive actions of the positive ions in the cytosolic site that will like to push the ions back into the extracellular milieu. And at resting state, once that equilibrium of the equilibrium of sodium ions is reached, you get the membrane potential to be established. Now, in an animal cell, the membrane potential is established by potassium, and more importantly, by the leaky channels that allow potassium to leave the cell. So now, here in the left, in part A, we have the leaky potassium channels. This is a channel that opens and closes on its own, so it allows for the flux of potassium ions across the membrane. So if the leaky potassium channels are closed, the plasma membrane potential will be zero, because there is no exchange of positive and negative ions between the two sides of the membrane, and therefore the charges are equal exactly and balanced. But when the potassium leaky channel opens, that membrane electrochemical gradient is going to drive the potassium ions to move from the high concentration of potassium in the cytosol to the low concentration of potassium outside. But that also is going to be countered by the driving force due to the voltage gradient. As you know, as you can appreciate, more positive ions are concentrated in the outside part of the membrane, and more negative charges are concentrated in the inside part of the membrane. So whereas the potassium gradient, it's driving the potassium ions to leave the cells through the channel. The membrane potential, the voltage gradient, on the membrane potential, it's attracting the ions inside the cell because of the greater negative charge within the cytoplasmic part of the cell. So, in that regard, the membrane potential exactly balances the tendency of the potassium ions to leave due to their concentration gradient. This is also illustrated in this other figure. So this other figure is showing the potassium leaky channel here in purple. And as you can appreciate, inside the cell there are proteins which are also charged. So these proteins have negative charges over here. The membrane charge in the cytoplasmic region of the membrane, it's negative, and the membrane charge in the outside of the membrane, it's positive. So the resting membrane potential is established when the movement of potassium ions out of the cell is going to equal the movement of potassium ions into the cell. And we have two forces driving that movement. The first force is the potassium diffusion down its concentration gradient outside of the cell via the leaky channels. So the loss of potassium results in a negative charge in the inner side of the plasma membrane. But because now we have an inner negative charge inside the plasma membrane, those potassium ions are going to be attracted back into the cytoplasm to that negative charge, and therefore they will go back through the channels inside the cytoplasm. So therefore, the movement of ions outside of the cell, the movement of potassium ions outside of the cell, it's balanced by the movement of ions, of potassium ions, back inside the cell. One force drives the concentration gradient of potassium, drives the exportation of the ions outside of the cell, and the negative charge created by the membrane potential attracts the movement of those potassium ions back inside the cell. And that generates a resting membrane potential that is about minus 60 millivolts. Now, we can use a simple mathematical equation called the Nernst equation to calculate the membrane potential of a cell, but only if the ions are single positive charge ions. So now, the force that tends to drive the ions across the membrane, as we know, has two components: the electrical membrane potential and the concentration gradient of the ion. And as I mentioned, when those two forces are at equilibrium, the forces are balanced, and basically they satisfy the relationship given by the Nernst equation. Basically, that the voltage in millivolts is going to be equal to 62 log 10 of the product of the outside concentration of the ion divided by the internal concentration of the ion. So depending on, and that is going to be the part that establishes the gradient. What is the concentration of ions in the outside divided by the concentration of ions in the inside? So this will then give you the value of the membrane potential in millivolts. And we can then use that to calculate the resting membrane potential, but only for single positive charge ions. Now, let's take a moment to look at how ion channels in nerve cells were studied. So we're going to take a moment to look a little bit about nerve cell anatomy. We're going to see how Loligo Pillay, the squid, is used as a model organism to study ion channels in the nerve, and how the voltage-gated ion channels in the surface of the neuron cells are able to allow the movement of ions across the membrane to change the membrane potential of the cell. That in turn is going to generate an action potential that could be propagated down the axon of the neuron. And last, we're going to explore how those electrical signals eventually get converted to chemical signals that the neuron can release to activate other cells. So when we look at neurons, neurons are specialized cells that are designed to transmit information along the central nervous system or the peripheral nervous system. So a neuron will have a cell body, over here shown where the nucleus is, In the cell body, you have many dendrites, which allow that cell to have and receive information from adjacent neurons. And from the cell body, you have the extension of a membrane as a very long appendage called the axon. At the end of the axon, you have the axon terminal that is also separated into small protrusions that could then interact with cells through the membrane in a synapse. That will be an example of a multipolar interneuron. Other neurons have different anatomy. In the case of a motor neuron, the motor neuron also have a cell body, different types of dendrites, and of course it has an axon that facilitates the movement of the signal from the cell body all the way down to the axon terminal. The difference is that the motor neuron has myelin sheaths, which are membranes that are wrapped around many times around the axon. Those are usually, depending on the peripheral nervous system, Schwann cells, or in the central nervous systems from oligodendrocytes. But what those myelin sheaths can generate are insulation for the axon, so it can help propagate the signal a lot faster. In between the myelin sheath, you have small areas of the axons which are exposed, about a micron in length, and those are called the nodes of Ranvier. Most of the time in class, we're going to concentrate on an unmyelinated axon, like the one shown over here in the multipolar interneuron, and we'll not really spend a lot of time looking at the myelinated axon in the case, for example, of the motor neuron. So most of the material that we're going to cover and examples are going to be of the unmyelinated axon, for example, in the multipolar interneuron. At the axon terminal, shown here by this transmitted electron microscope, we have contacts of the axon terminal with a postsynaptic cell. In this case, the postsynaptic cell is a muscle cell. So this is going to be a motor neuron that is going to be able to extend its axon all the way to the muscle and have an interaction at the neural muscular junction. At that point, in the axon terminal, we have the area called the synapse, where the membrane of the neuron touches, or it's at close proximity with the membrane of the muscle cell. Now, inside the membrane of the axon terminal in the neuron, we have many small vesicles shown here as the circles, and those are called synaptic vesicles. Those synaptic vesicles contain neurotransmitters that, when a signal from the neuron travels across the axon and gets to the axon terminal, they will be released into the space next at the synapse to stimulate the muscle to do a response. And that is the area which we consider the basal lamina in the synaptic cleft. What you can also appreciate from this image is that the plasma membrane of the muscle tends to invaginate and goes inside the cytoplasm in a structure that later we'll learn that is called AT-tubule. Below, in the muscle cell, you have a structure of reparative fibers that make the sarcomere, which is the muscle contractive proteins, and those are, we're going to learn later, that those are actin filaments with myelin fibers that will contract once the neuron has given the signal to that muscle cell to contract. Now, Loligo, the squid, has been used as a model to study nerve cell potentials for many years. As you know, the squid is a very fast arthropod, so it's actually a cephalopod from the same family as octopus, and it is able to move really quickly in water by propulsion of water through its body. It has an excellent vision system and is able, therefore, to hunt really effectively with its tentacles. Because of this, the squid has what is considered one giant set of neurons to convey information from its brain to its muscular body. And that giant axon, it's actually very large compared to the axons that we have in mammals. The larger the diameter of the axon means that the information can travel faster. So what scientists are able to do is that they're able to isolate the giant axon of the squid and put an electrode inside of it, and being able then to measure the membrane voltage across the membrane. So as you can see here in A, you have the electrode going inside. You have another electrode placed in the buffer of the media that you're having the axon, and you have a little potentiometer to measure voltage. The axoplasm, it's another word for the cytoplasm inside the axon. And therefore, once you can apply a stimulating signal to the membrane, you can then record electrical signals as the ions are flowing into the giant axon. So let's learn about what is a depolarization. Membrane depolarization, it is a change in a cell's membrane potential that is going to make that membrane potential more positive. In neurons and other cells, if you have a membrane depolarization of a particular magnitude, that can pass a threshold that will result in an action potential. So that threshold needs to be overcome before the action potential can happen. And we're going to look at how the action potential looks when you're measuring it in a potentiometer. On the other hand, you can make the membrane more negative by hyperpolarizing it. So that will be the opposite of depolarization, and by making it more negative, you can inhibit the rise of an action potential. So let's look at the action potential that we can measure with a giant squid axon in the lab by using a potentiometer. As I mentioned to you earlier, if you're having the connection inside the cytoplasm of the axon and you have another electrode in the outside, you can give and measure the membrane potential at resting points. And as you see here in this line, that membrane potential, it's usually around -60 millivolts. Now, if you give a brief electrical stimulus to the membrane, that brief electrical stimulus to the membrane will make the membrane more positive. So as you can appreciate from this depolarization stimulus, the membrane potential went from -60 to -40, being more positive. Now, in a membrane that doesn't have channels, the potential change in membrane potential will eventually reset back to the resting state as shown here in the green line. But in a membrane that has channels, that initial depolarization stimulus that has taken the membrane potential from -60 millivolts to -40 millivolts will have effect in the voltage-gated sodium channels on the membrane, and that change in voltage potential will make those voltage-gated sodium channels to open. When they open, you're going to have an influx of sodium ions inside the cytoplasm, and that will result in an increased positive charge within the cytoplasm, eventually passing the membrane potential from -40 to 0, and later reaching plus a little bit over +40 millivolts to what is called the equilibrium potential for sodium ions. We're going to see that increase, rapid increase in membrane potential, it's called the action potential. And the action potential, it's generated by the depolarization signal given to the neuron cell. So let's see how that is happening inside the neuron cell. So here in the upper part of the image, what we have is a membrane showing the concentration gradients of sodium and potassium. And as you can appreciate, you have about 150 millimolars in the outside for sodium and 112 millimolars, for the concentration of sodium inside the cell. Potassium, as we have discussed, has a concentration gradient that is greater inside the cell than outside of the cell. So at resting stage, the potassium leaky channels are open, and that allows the migration of potassium ions outside of the cell. Since you have a negative charge in the inside of the cell, that also will attract some of those potassium ions to the inside, where the until it reaches equilibrium. Basically, the number of ions leaving the cell equals the number of ions being pulled inside by the membrane potential. But what you can see also is that you have a series of sodium channels called voltage-gated sodium channels, which are closed. Now, when you give a depolarization stimulus, what you start to see is that those voltage-gated sodium channels, once you experience a change in membrane polarity, those channels will open. So appreciate that now, once the channel opens, you're going to have an influx of sodium ions, and that influx of sodium ions are going to change the local environment in the cytoplasm area around that channel, making now the inner part of the membrane more positive and the outer part of the membrane more negative. And that is when you begin the depolarization state, which eventually will lead to a change in voltage within the cell called the action potential. So let's take a look at those voltage-gated sodium channels, because they are important in maintaining the membrane action potential. So at rest, when the membrane of the cell is at rest, these voltage-gated sodium channels are closed. Again, notice that you have a positive charge in the outside of the cell and that you have a negative charge, in the cytosolic part of the cell. In this illustration, part of the protein of the voltage-gated sodium channel is positively charged. Now, when you have a depolarization event, and that depolarization event results in a change in membrane potential, now you have more positive charges in the inside of the membrane and more negative charges in the outside of the membrane. At the arrival of this action potential, because of that depolarization event, the channel opens up, responding to the change in voltage. Illustrated in this image, the red rectangles attached to the channel will be attractive to the negative charges in the outside of the cytoplasmic membrane, and therefore open. But this channel has a third state, and that state is an inactivated state. That even if the membrane is depolarized and the channel has been open because you have more negative charges in the outside than positive charges in the inside, a portion of the protein is able to block the channel and therefore prevent the movement of ions across that channel. So therefore, in this inactivated state, the channel is opened, but because the protein component has now moved to block the channel is inactivated. Eventually, When the membrane becomes repolarized and the charges move to be more positive in the outside and more negative in the inside, the channel will reset and go back to the closed configuration of the channel. So again, the channel is moving from three different stages depending on the membrane potential. A closed state, when the membrane is at rest and therefore having more positive charges in the outside than the inside. An open state, where the channel now opens because you have more negative charges in the outside than the inside, and this allows the flux of sodium ions inside the cytoplasm. And an inactivated state, where the channel is open but blocked, and therefore you stop the influx of sodium ions inside the cell. When the membrane is repolarized, the channel goes back to its closed conformation. So you have a channel that is able to move back and forth between these three different stages. So let's look at how the stages of the channel are reflected within the action potential. So here at the beginning, we have the cell at rest, and therefore you have an action membrane potential, you have a membrane potential of about minus 60 millivolts, At that point, the voltage-gated sodium channel is completely closed. At time zero, shown over here in the upper part of the graph, we have the stimulating current. And we can give a very brief stimulating current that is going to induce a depolarization of the membrane. If that depolarization reaches a particular threshold, that is going to induce the voltage-gated sodium channels to open. When they open, they're going to allow the influx of sodium ions inside the cytoplasm, and that is going to result in the action potential becoming greater and greater and moving from a negative membrane potential to a positive, because you have positive ions moving inside the cell. But when it gets to a particular positive range, over here close to the equilibrium constant for sodium, the channels become inactivated, and therefore no more sodium ions are able to enter the membrane, are able to cross the membrane and enter the cytoplasm. And at that point, you begin to reset the membrane potential with time until it resets back to the resting phase and the channels close back again, moving from the inactivated state to the closed state. One thing that I would like to point to you is the setting up of the decrease of the membrane potential. What we have, it's this reduction in membrane potential from more positive going back to the resting state, which is more negative, and that is called the refractory period. So how is it happening that within a couple of milliseconds, the membrane potential can go from positive 40 to about minus 40? This is the effect of having now voltage-gated potassium channels that are going to be able to open when the membrane potential is very positive. Those, unfortunately, are not shown over here in this image. But let's just imagine what will happen. So as we discussed earlier, at the resting state, the membrane potential is around minus 60 millivolts. When you have a depolarization signal here at time zero, that depolarization signal may be sufficient to induce an action potential, and the membrane potential changes to a bit more positive of about minus 40 from minus 60. That induces the opening of the sodium voltage-gated channels, which are going to allow the influx of sodium ions into the cytoplasm, and that increases the membrane potential from minus 40 to around 40 millivolts. At that point, of around 40 millivolts, you have now the voltage-gated potassium channels opening up, and they're going to now allow the efflux of potassium ions from the inside of the cell to the outside of the cell. That then results in a decrease of positive ions within the cytoplasm of the cell, which is going to result in a great and rapid decrease of concentration of positive ions within the cytoplasm, resulting in the resetting of the membrane potential back to the resting state. You also are going to have the potassium-sodium antiporter pump that is going to spend ATP to bring back more potassium ions to the inside of the cell and pump out sodium ions to the outside of the cell, reestablishing the gradient. But this fast refractory period results from the opening of the voltage-gated potassium channels that are going to release the potassium ions into the extracellular milieu, resetting back the membrane potential to the resting state. Now, how is the action potential propagated? What I'm going to show you is this image over here that is illustrating the membrane of the axon full of small voltage-gated sodium channels. The voltage-gated potassium channels are not shown in this image. So now, let's look at this area in blue or light blue, that it's the initiation of the depolarization. So at a certain point, at time 0, when you have triggered the action potential will induce a depolarization, and as you can see with this little voltage-gated sodium channel, that is going to induce the opening of that channel to allow the influx of sodium ions into the cytoplasm. That results in the propagation of a positive charge within the cytoplasmic part of the membrane. Those ions are able to disperse rapidly to the right and left of the ion channel that is open. But because the ion channels on the left are inactivated, they are not able to allow sodium ions to cross the membrane into the cytoplasm. But the ion channels that are in the right of that initial open voltage-gated sodium channel are closed. And therefore, as the sodium ions are rushing in and sort of dispersing to the local environment, they're able to depolarize the membrane, allowing now for those neighboring closed voltage-gated sodium channels to open. That will result then in the influx of the sodium ions inside the membrane, so inside the cytoplasmic area, with the concomitant inactivation of the channel who was previously open. So at 1 millisecond, what you can appreciate is that the depolarization event has propagated to the right of the axon, leaving behind to its left some inactivated voltage-gated channels that are not able to allow influx of sodium, and much farther down the axon, channels which were previously inactivated but that have now reset to the closed configuration and therefore will be able to respond to a depolarization signal that comes to them. But because of the nature of the inactivation of the channels, the polarization and the action potential only migrates into the direction of the axon to the right. So that prevents the propagation of the action potential to the left. The fact that the voltage-gated sodium channels are inactivated. In this image from the molecular cell biology book, it's showing the same event, showing little voltage channels, but it's also showing you the distance along the axis and the membrane potential as it is changing with the movement on the axis. So what you can appreciate from here, for example, at position 2 millimeters, you have the voltage-gated sodium channels which are open, and therefore allowing the ions of sodium to come inside. They're going to induce a depolarization to the right, therefore allowing the channels which are at position 3 millimeters to open up, and also allow sodium ions to come inside. The voltage-gated sodium ions which are on the left at position 1 millimeter are closed and are inactivated, and therefore they're unable to allow sodium to come inside the axon. Therefore, the membrane potential gets moved only to the right in the area where you have bona fide closed channels. So at time 0, the depolarization event happening around 2 millimeters in the axon, it's able to move by 1 millisecond to the position 3 millimeters, and by 2 milliseconds to position 4 millimeters, leaving behind a group of inactivated channels that are not able to allow sodium ions to come inside. And therefore, the signal gets propagated to the right of the axon, away from the initial site of depolarization. This could also have been shown when you install different potentiometers along the axis of along the length, of the axon. So in this experiment, the scientists put 3 different voltage meters, one at the place that they call 0, one at the place farther down the axis, and a third one at a different position farther down the axis. And what you can appreciate is that the potentiometers are able to measure voltage where the initial action potential is generated, but with time that voltage goes down and is only be able to be detected by the voltage meter 2, because the membrane action potential, it's traveling to the right, and eventually that becomes 0 again. And the signal is being able to then be detected by the voltage meter 3 as a wave that is moving from left to right along the axon. So that's the process of propagation of the action potential. Now, let's look at this explained by a movie.
The fundamental task of a nerve cell is to receive, conduct, and transmit signals. Neurons propagate signals in the form of action potentials, which can travel great distances along an axon without weakening. To transmit an action potential over such a distance without weakening requires that the signal is continuously reamplified along the way. The central molecular players in this process are the voltage-gated sodium channels, which undergo a cycle of finely choreographed conformational changes. When an action potential passes, sodium channels open in response to membrane depolarization. Sodium ions rush into the axon, further depolarizing its membrane. Within a fraction of a thousandth of a second, however, the sodium channels switch to a new inactivated state, in which they are closed, but now also refractory to reopening. In this way, the membrane potential can recover quickly after an action potential has passed. The sodium channels then reconvert to the closed state, ready to be opened again when the next action potential is encountered. Let's examine the changing state of the sodium channel during an action potential. When no stimulus is present, the sodium channels remain closed, and the electrical potential measured across the membrane remains constant. However, if a depolarizing stimulus is applied by a brief pulse of electric current, the membrane will start to depolarize, away from the resting potential of about -80 millivolts. Some of the sodium channels will open, permitting sodium ions to enter the axon along their concentration gradient. If the depolarization is sufficient, even more sodium channels open, and the membrane potential rapidly approaches the equilibrium potential for sodium, about +40 millivolts. At this point, the sodium channels close, adopting the inactive conformation, where the channel is unable to open again even though the membrane potential is still depolarized. The sodium channels will remain in this inactivated state until a few milliseconds after the membrane potential returns to its initial negative value. The action potential is propagated along the length of the axon in only one direction. By examining the membrane potential and state of the sodium channels along a length of the axon, we can see why this is so. As a depolarizing stimulus reaches our section of the membrane, sodium channels open and current flows into the axon. This in turn depolarizes adjacent sections of the membrane, causing adjacent sodium channels to open, and the action potential is thus propagated along the axon. Sodium channel inactivation prevents the depolarization from spreading backward along the axon.
All right, so now with that image, we understand that when there is a depolarization event, that depolarization event is going to make the membrane potential a little bit more positive, and that is going to influence the voltage-gated sodium channels to open up. When they open up, they're going to allow for the influx of sodium inside the cytoplasm, which is going to greatly then generate the action potential, which is going to force other nearby voltage-gated sodium channels to open as well. At a certain voltage, about plus 40 millivolts, those opened voltage-gated sodium channels are going to be assuming the inactive form, where they will be open but not able to allow the movement of ions inside the cell. As the membrane is polarized, the action potential gets propagated in the direction of the axon because it is influencing the opening of closed voltage-gated sodium channels. Now, let's take a look at what happens downstream the axon, where the axon now has reached its target, the postsynaptic cell, and that electrical signal gets to the end of the cell. So what we have in this image are a nerve terminal, so a axon terminal, and inside the axon terminal we have a lot of small vesicles present. That axon terminal is interacting with the membrane of another cell. Since this is a synapse in the nerve, we call this the presynaptic nerve terminal. The cell that is interacted is going to be the postsynaptic cell, and it could be a nerve cell, it could be a muscle cell. So it could be any kind of cell that is going to be interacted. We always call it postsynaptic cell because it's interacting with the synapse of the nerve cell. Now, in the presynaptic cell, in the nerve terminal, we have the membrane. That membrane is going to be interacting with the postsynaptic membrane in what is called the synapse, and the synapse it's full of small vesicles, and those small vesicles are called synaptic vesicles, and those contain neurotransmitters. Now we have different kinds of neurotransmitters. We have some neurotransmitters who can excite neurons. An example of that will be acetylcholine, glutamate, dopamine, and norepinephrine. But we also have other neurotransmitters which are inhibitory. The amino acid glycine is a neurotransmitter that is inhibitory, and the gamma aminobutyric acid, or GABA, is another inhibitory neurotransmitter. Histamine and serotonin and epinephrine are also excitatory neurotransmitters. So depending on the neuron that that we talked about, they may be able to release a excitatory neurotransmitter or an inhibitory neurotransmitter. Now, in the postsynaptic cell, in the case of a muscle, for example, we have a acetylcholine receptor, and the acetylcholine receptor is a neurotransmitter-gated sodium channel that is able to bind to acetylcholine, which is its ligand, by having binding sites to it, and therefore convert that chemical signal released from the neuron as acetylcholine into an electrical signal. So the acetylcholine receptor, the neuramidic acetylcholine receptor, because there's more than one type of acetylcholine receptor, contains 5 subunits, and all the 5 subunits are able to, some of those 5 subunits are able to bind to acetylcholine. Usually, in the absence of acetylcholine, that channel is going to be closed. But when the acetylcholine molecules, 2 of them, bind to the channel, they're going to induce a conformational change that is going to open the channel, allowing sodium ions to move inside the cell. And that is called the open configuration. So let's take a look at this combination of information from the neuron perspective and from the postsynaptic cell perspective. So now, at a resting neuron terminal, we have the axon terminal, and the axon terminal, as we mentioned, has postsynaptic, presynaptic vesicles full of neurotransmitters. But they also contain a different kind of voltage-gated channel. This one, it's a calcium-gated calcium channel. So when open, it will allow the influx of calcium ions. In the synaptic cleft, meaning the space between the neuron and the postsynaptic cell, we have in that area, in the postsynaptic cells, neurotransmitter receptors on the surface of the cell. So when you're having an action potential migrating down the axon, and that action potential gets to the terminal, it's going to induce the opening of the voltage-gated calcium channel, and that is going to allow for the influx of calcium ions. The influx of calcium ions are going to mediate the fusion of the synaptic vesicles, which then will release the neurotransmitters into the synaptic cleft. Those neurotransmitters will then be able to bind the neurotransmitter receptor. So this is how an electrical signal that is traveling down the axon of the neuron will be converted to a chemical signal, be that the chemical, the neurotransmitter. All right. So after the release of the neurotransmitters into the synaptic cleft, the neurotransmitters will be able to bind to the neurotransmitter receptor on the surface of the postsynaptic cell. That is going to allow the channel to open and therefore have an influx of ions inside the cell. That influx of ions inside the cell will induce a depolarization of the membrane of the postsynaptic cell, and that depolarization will result in an action potential. So now you can see that the chemical signal that has been released by the neuron can be converted back into an electrical signal by the binding of that neurotransmitter to the activated receptor, which will then lead to the generation of an action potential in the postsynaptic cell. This can be represented here by this other movie.
Neurons transmit chemical signals across synapses, like the one shown in this electron micrograph. We can identify the dendrite of the receiving or postsynaptic cell, as well as 2 presynaptic nerve terminals loaded with synaptic vesicles. Note the narrow cleft separating the pre- and postsynaptic cells. The synapse converts the signal of the action potential in the presynaptic cell into a chemical signal. When an action potential reaches a nerve terminal, it opens voltage-gated calcium channels in the plasma membrane, allowing calcium ions to flow into the terminal. The increased calcium in the nerve terminal stimulates synaptic vesicles to fuse with the plasma membrane, releasing their neurotransmitter cargo into the synaptic cleft. The released neurotransmitters diffuse across the synaptic cleft, where they bind to and open the transmitter-gated ion channels in the plasma membrane of the postsynaptic cell. The resulting ion flows depolarize the plasma membrane of the postsynaptic cell, thereby converting the neurotransmitter's chemical signal back into an electrical one that can be propagated as a new action potential. The neurotransmitter is quickly removed from the synaptic cleft, either by enzymes that destroy it, or by reuptake into the nerve terminals or neighboring cells.
So now, not all receptors are going to be excitatory. Some of them are going to be inhibitory. So some of these neurotransmitters will induce membrane depolarization, while others are going to hyperpolarize the membrane, preventing depolarization. For example, the acetylcholine receptor, it's one which, when acetylcholine binds to the receptor, it will open the channel, allowing sodium ions to come inside. That influx of the sodium ions is going to depolarize the membrane, increasing the likelihood of an action potential being generated. Remember that there's also always a threshold that needs to be surpassed to generate that action potential. On the other hand, the activation of an inhibitory channel will do the opposite. So, if in the case of the GABA receptor, when GABA binds to the channel, it's going to open up, allowing the influx of chlorine ions. The chlorine ions, being negative, are going to move inside, and it's going to keep the membrane depolarized because you're increasing the amount of negative ions in the cytoplasm. That will decrease the likelihood of having an action potential being generated if you have a depolarization signal. Because remember, the depolarization signal, it's making the area within the membrane more positive. So an inhibitory neurotransmitter like GABA is able to then maintain the membrane in a more negative state, hyperpolarizing it. In this table, you have a list of the ion channels which are present in the neuron cells, and where are they present and what is the function. So I'll encourage you to look at a lot of these channels and understand what is their function, if they are excitatory signals or if they are inhibitory signals, and where they are located. So let's take a look at what's going to happen in an overall signal. So in this case, we have a motor neuron cell that is having an action potential being propagated along the axons until it gets to the axon terminal. At this point, in this presynaptic cell, you're going to have that action potential coming over the membrane, and that is going to induce the opening of voltage-gated calcium channels that are going to allow the influx of calcium to the localized cytoplasm. The influx of the localized calcium, it's going to help fuse the synaptic vesicles that are present in the synapse terminal to the membrane, and therefore that is going to release acetylcholine present in the synaptic vesicles into the synaptic cleft. The synaptic cleft, it's the area where the neuron cell and the muscle cells are interacting, and therefore the acetylcholine being released, it's going to be able to bind to the nicotinic acetylcholine receptor. When it binds to the receptor is going to induce a conformational change in the channel, opening the channel, and therefore you have the influx of sodium ions into the muscle cell. That influx of sodium ions into the muscle cell will induce a localized depolarization, and being strong enough, that is going to induce an action potential. That action potential will then migrate down the membrane of the muscle cell, and it's going to be propagated by a very similar mechanism involving voltage-gated sodium channels. So therefore, they're going to open up, allowing more sodium ions to come inside and propagating the action potential along the surface of the muscle cell. Now, the muscle cell, as I mentioned to you earlier, has some invaginations in its membrane, and those invaginations in the membrane are called T-tubules. Those invaginations of the membrane are going to come deep inside the cell, and they can come very close to the cytoplasmic reticulum of the muscle cell. And as I mentioned earlier, the sarcoplasmic reticulum in a muscle cell, it is the endoplasmic reticulum of that cell. So now, within the T-tubules, as the membrane action potential is traveling down the membrane inside the T-tubule, it's going to induce the opening of voltage-gated calcium channels within the T-tubule, and that is going to interact with some channels in the surface of the sarcoplasmic reticulum, allowing calcium to come in from the sarcoplasmic reticulum into the cytoplasm. That great influx of calcium ions within the cytoplasmic region of the muscle channel, because the cytoplasm, it's being inundated by calcium ions that are fluxing from the cytoplasmic reticulum, will induce muscle contraction. In later lectures, we're going to then see how calcium ions that are released from the sarcoplasmic reticulum will result in muscle contraction. But at this point, what I want you to emphasize is the fact that an electrical signal that is coming down the motor neuron will be converted into a chemical signal in the form of acetylcholine, and then how that chemical signal, when it binds to its receptor, the nicotinic acetylcholine receptor, will be converted back to an electrical signal that will be propagated as an action potential down the muscle cell, and therefore result in the release of calcium from the sarcoplasmic reticulum, making the muscle contract. Eventually, the muscle needs to relax, and part of that relaxation will involve the pumping of that calcium from the cytoplasm back into the lumen of the sarcoplasmic reticulum. And as we mentioned earlier, that will be done by the calcium pumps that are going to use ATP hydrolysis to pump back the calcium inside the lumen of the sarcoplasmic reticulum. So to finish, I have put some resources that I would like you to explore to get more information. You have the neuron action potential being explained in this website. You have a website from Columbia University explaining neural signal, and you have a small animation on action potentials from the SYNAPSE website, as well as a good explanation of it from Wikipedia. So this lecture will be more covered with the flip lecture on Monday. So I will wish you a good weekend, and this will be the last lecture that will be covered in the exam. Thank you, and see you Monday.